Hi, my name is Ishan, and today I'm going to talk about the GFP gene and its incubation in mammalian cell line HEK293 to study the cellular processes and the protein dynamics. So the here we can see is the aim of the project as expressing the GFP gene in mammalian cell for visualization of several protein expressions and the transfection efficiency as well. The gene of interest is going to be the GFP gene. The expression vector is the pcDNA 3.1, supported with the picture below it, and the host cell, which is the mammalian cell line HEK293, which is also known as kidney cells. Then comes the GFP mechanism, the green fluorescence protein. So GFP undergoes automatically formulation of fluorescent chromophore within the protein. So the chromophore formulates and allows GFP to emit its green fluorescence. So it enables non-visible images in the real time of biological processes in living cells and organisms. The next slide is going to be the steps involved in GFP cloning. So as I have listed the steps as well with the diagram supporting saying the number and the steps which take place over here. So GFP gene is undergoes isolation. After isolation, PCR amplification. After PCR amplification, it is transferred into a plasmid vector. Then the vector is transformed to a bacteria known as E. coli. After all this, plasmid undergoes a purification process where it is purified and then transmitted into a mammalian cell. And then the last step is selection and screening, where identified cells are identified by incorporating GFP gene plasmid using selection markers, and this process method is also called known as fluorescence. Now here we need to transform it. Here we can look at the bacterial transformation of GFP in HEK293 mammalian cell. The steps are as follows. First, it goes under isolation, then amplification by PCR to make multiple copies. After making multiple copies, plasmid vector goes under cloning, where GFP gene is inserted in a circular plasmid as well. Then the transformation of gene into bacteria, where recombinant plasmid is introduced into typical E. coli, where then plasmid is purified again, then transferred into HEK293 cell, and it is identified by green fluorescence. And the next is QC validation, where we need to follow the flowchart as given below. First, grow the cell in the labs, then extract RNA, then convert RNA to uh, cDNA, and the next step where we need to run the qPCR for analysis, and after performing the qPCR, the cDNA to measure how GFP gene is being expressed, the analysis, the result undergoes certain expression levels. After we obtain the results are going to be here. I've explained the transfection process, how things are like to analyze the gene expression and the protein localization, as well as with including cellular processes in the mammalian host cell. So here the results are the fluorescence observation, where we can see the transfection cell will be expressed in GFP, making them visibly in the fluorescence under fluorescent under fluorescence microscope, where basically this green fluorescence is a direct indication of success transfection and GFP expression. So the non-visible organelles are going to be seen in the microscope, and here we can see this is then the experiment is done. Yeah, thank you.